Thank you. All right. Nice to see you all. Now, I'm going to be the first one to tell you that my wife will tell you I am terrible recognizing faces. Well, let's say this. I recognize people, but I can't remember who they are. So if you were at the meeting last year, just come up and say, nice to see you again. I'm blah, blah, blah. And then that'll trigger something. But I do this with people I work with. I run into them at the mall or something, and I can't even recognize who they are. So forgive me. It's not that I don't care. I just don't have the capacity. But any case, one of the things I wanted to talk to you guys about is how many of you are new families to the meeting this year? If you could just raise your hands and stuff. Okay, so this is a talk that just is kind of explaining how to interpret your genetic report, and then it goes a little bit into the disorder and stuff like that. It's by no means something I'm expecting you guys to memorize, but at the same time, what I'm thinking is maybe later on in the day, those of you that are new, we can all kind of get together somewhere and write down your questions and stuff like that you might come across here, and we can go over things. Now, the other thing that I do when I come to these meetings is I try to recruit patients into my studies. And right now, you'll find tomorrow, we're kind of in between a couple studies, but we are looking for some children that have a particular type of variant. They're called polygenic variants, where it's a large deletion involving a couple of genes. And we're also looking for parental mosaicism, but I'll get into that tomorrow. But if that's the case with you, we can talk about enrolling you and everything. Now I have to close this out. Now, this is not supposed to be, biology 101. It may come across that way, but what I'm trying to do here is set the stage so that you guys understand a little bit of the genetics behind all this, and then this is going to segue into our research work that we'll talk about tomorrow, and hopefully that will kind of grease the grooves so the understanding's a little bit there. I know families who were here last year, this is going to be very similar to the first talk I gave last year. I've been giving this talk since St. Louis, which is probably 8 years ago, if not more. And families tend to tell me every time they hear it, they learn a little more, it becomes a little bit more understandable to them. So forgive me if it's something you've seen before, but hopefully there's some things to glean from it. In any case, this talk is called Understanding GAND and the Genetics Behind GAND. I'm a pediatric neurologist in the departments of pediatrics and neurology at Cedars-Sinai in Los Angeles. I'm also an associate professor at UCLA, and I'm also a part of the Board of Governors Regenerative Medicine Institute in GARD Children's at Cedars-Sinai. So this was the first case of GAND I ever met, and she was 20 months old. And let's see, yes. Can you guys see that? Or maybe I need to use the laser pointer. Okay, there we go. That's a little better. So she came into my clinic. She was 20 months old. She had hypotonia. She had no speech, and she had some motor delays. She had a large head and had what's called frontal bossing, which is a little bit of protuberance of the forehead, as well as hypertelorism, which is kind of widening between the eyes, and a particular type of nose. And she also had bicuspid aortic stenosis, which is a heart valve defect. And then she needed to have a procedure at 1 month of age called a valvuloplasty. Now, we figured out that about 8% of the kids with GAND have bicuspid aortic valves, and we recommend to everyone that their child see a cardiologist to double check that. Now, usually it's a benign thing that they live with, but at the same time, it needs to be followed. On top of this, on MRI, she had what we call extraaxial fluid collections, as well as large ventricles. And then this is a structure called the corpus callosum, which means crossing fibers. And so the fibers that went from one side of the brain to the other side were less pronounced. So when we saw her, she had a clinical diagnosis of motor language delay, diffuse hypotonia, macrocephaly, aortic stenosis, bicuspid aortic stenosis, and these distinct facial features, which at the time, this was about 2016, we really didn't have a clue what this could be, but we had some techniques that allowed us to send off a screen. It included the child and the parents, and we got back a GATA-D2B variant. And her variant was a de novo, meaning it was new to her and the family, meaning that neither parent carried it. And it was what we call a nonsense variant, which, just so you understand, hopefully by the end of the talk, you'll understand this and this a little bit more, but this is what's called the cDNA variant. And this is the actual variant on the messenger RNA that encodes the protein. And this is the translation of that variant on the protein. And basically what this says is a glutamine at position 204 is replaced with a stop codon. Hopefully, in about 45 minutes, you might understand what that means if you don't already. So before we get into all that, I just want to kind of explain how DNA is organized in the cell. So, we all know this kind of double-stranded helix that consists of A's and T's binding to each other, which are the red and the purple, and the C's and G's, which are the yellow and the green here. But that strand can't just kind of hang in the wind. It needs to be kind of anchored down. And so you have these proteins that are called histones, which they form an octamer and another octamer, and the two octamers come together. No, I said that wrong. There's four of them, and they come together to make an octamer. And so these nucleosomes have two wraps of DNA around each one, and you can have it where it's open, or you can have it like, in the biology pictures of cells dividing, you can have very condensed chromatin that makes up chromosomes. Okay, so this is an inactive form of DNA, and this is a more active form of DNA. And we're going to get into that a little bit more over the next couple of days. So if we take this strand of DNA, what we can see is that there's multiple genes that are aligned on a single strand of that. And some of these genes are turned on in all cells. So if this is gene D, it's in this orientation, and this is what we call promoter, this bent arrow, and that says that that's where the gene starts to be expressed. Now, this promoter is such that it's active in all cells. Now, if we go to gene C here, we've got a promoter that's brain specific. So this gene is turned on in brain cells. And if you're a liver cell, this promoter is going to get shut down. And the same here, if you're a liver cell, this promoter is going to get turned on. Now, the essence of what we're going to be talking about over the next couple of days is exactly that, is how does a gene get turned on and activated or turned off and repressed in different cells, specifically in the brain, okay? So if we take this, and unfortunately to get there, we need to go through a lot of this biology 101 stuff. So here's that same locus of DNA with those four genes, and we're gonna look at this brain-specific gene a little bit more. And when you look at it, it's organized into, again, this promoter, and then the coding sequence for the protein. But the coding sequence consists of what are called exons, which actually code for the protein, and introns, which are called intervening sequences, which are just spaces there. Now, you might go, why do you need that? And one of the ideas is, viruses integrate into DNA, so if you make the coding sequences 1% of your DNA, then the viruses can integrate into the 99% that's left over and not affect the genes that need to be functioning. So this is the main blueprint. So I always use analogies of building buildings. So you have an architect's office there, or a draftsman's office, and they're making a blueprint for making a building, and this is it. And then they make what are called working blueprints, which are the messenger RNA. And in messenger RNA, what happens is you make another strand of nucleic acid. This time you replace the T with a U, and then you take out those intervening sequences, so all you have is that messenger RNA there. And that's the essence of what the COVID vaccine was. They were injecting this messenger RNA into you, and the idea was that you could then translate that into the building, which is the protein, and then the messenger RNA goes away, which it typically does, but the DNA sticks around. So you're constantly making these working blueprints. Now, the building is the protein here, and it's built up of 20 different amino acids. And instead of going and saying, this is alanine, and this is leucine, and this is glycine, and things like that, I'm just using different shapes. And so these 20 different shapes can be put into a chain, and each chain has a little bit different chemical properties. And so some of it's the keratin that's in your fingernails, and others is, say, hormones that you're secreting from your liver or something like that. And then these chains can then form these complex 3D structures where they can have parts that are these helices themselves and form like a globule. And then those globules can form a bigger structure or a complex with other proteins. And that's important because that's what GATA-D2B does. It forms a complex, and that complex we'll get into is called the nucleosome remodeling and deacetylase complex, or the NERD complex. But that's down the road. Okay, so that's basic principles. Blueprint, working blueprint, building. So how do you get the diagnosis? Well, it used to be there was a number of different tests that you could use to figure these out. Now, SNP arrays have kind of gone by the wayside, and SNP arrays were basically tests that I'm going to use the analogy where it's kind of like putting little breadcrumbs along the DNA and then kind of washing the ones that don't stick to the DNA away, and then washing the ones that do stick to the DNA away, and then counting those up. And if some of those breadcrumbs didn't stick to a spot, then you go, well, that spot's probably deleted. Or if more of those breadcrumbs stick to a spot, then you say, well, that section was probably duplicated because there's more than there should be. SNP arrays were very valuable for looking at the structure of DNA. So you could see deletions or duplications. But by and far, these have kind of gotten, they've gone by the wayside. Now, people may still get these done, but because they're very cheap, but at the same time, you can answer those same questions with some of these other technologies down here. Now, a gene panel is basically a list of genes that they'll test, and those genes all have the same sort of presentation. So some of the panels would be for kids with muscle disorders, or some of them will be for kids with seizures, or some of them will be kids with intellectual disability. So the presentation is what then grabs, let's say 100 genes that they go, okay, all these 100 genes have presented with a muscle problem. And you send that off, and it will look at structural changes like deletions and duplications, but it'll also look at the sequence of all those genes and see if there's any that have been previously identified. And so they can say, oh, well, gene X has caused something very similar to what your child has, and there's 100 children who've had exactly that same variant in gene X. Now, the other thing that can happen is gene X can be here, and all the variants typically are over here, and you get a variant here, and then they go, well, this is on gene X, it's similar to what's going on, but because there's variants over here, we're not quite sure because it's usually over here. So then you might hear something called a variant of unknown significance. Now, I'm sure there's a few of you who got your genetic report back that talked about GATA-D2B, but also said, oh, and there's a variant of unknown significance in gene X or something like that. And we can talk a little bit more about that. Now, these bottom two here have kind of, glummed together, where exome sequencing is where they sequence the 1% of the genome that's the most important percent. So if we go back here, that's going to be just looking at these exons, all right? So that's 1% of the genome, so that's the coding section. So you're going to find about 80% of variants in that region. And so they said, hey, why don't we just look at that 1%? Now that was back when sequencing was a little more expensive and a little more difficult. Now sequencing is very easy, and so they can do 100% of the genome and get all these answers pretty quickly and a lot less expensive than it used to be. The only problem is they get 100% of the information, but they really can only look at the 1 to 3% that they already knew, along with some new stuff. Now you might go, well, why would anybody do that? What's the point? And the idea is you've got 100% of the information. Now it's stored in a record, and as the databases build up with information, you can start looking at the next 4%, 5%, 10%, until eventually we understand what's going on. Now, as I said, the exome is 1%, and 99%, the other 90%, 99% might just be junk stuff, and we don't quite know, but we're slowly picking things out and figuring things out. So basically, like, this is another picture. Exome is 1% of the genome. Genome is 100% of the DNA. These two modalities now can pretty much do all the structural findings where you can see deletions or duplications that the SNP arrays used to do. And as I mentioned, there's about 80% of disease-causing variants that thought to be in this 1%. Most of you probably have gotten exomes. A few of you probably got gene panels. And genomes, I'm not sure how available they are in the UK. They've just become pretty standard in the United States when we're sending things out. And, I always tell this to families from other countries and stuff. If American insurance companies are approving a study, that means these are the most stingy, miserly people in the world. So if they've said this is valuable, it means it's probably true. And so you guys should start putting pressure, if they're not doing genomes, start putting pressure on whoever you can to kind of say, hey, why aren't we doing this and stuff. So back to our patient. She had exome sequencing and had a de novo truncating nonsense variant. And as I said, this is that C610C to T. That's the messenger RNA, the working blueprint change. And then P stands for protein. C stands for cDNA, which is another way of saying messenger RNA, but I won't go into why you say one versus the other. P is for protein, and there's a glutamine at amino acid 204 that's been changed to a stop codon. So this is a nonsense variant. And a lot of the times we call this a truncating nonsense variant. And I'm going to show you a little bit of how this works. So again, you have the messenger RNA here, and it's made up of A's and U's and C's and G's. Now, for each one of these amino acids, each different shape here, there's a 3-letter code in the messenger RNA that encodes it. So AUG is always going to encode a circle, where UUC is going to do this trapezoid. I picked the hard one there. And then what we have is this one code that's UAA, which is a stop codon. So that tells the cell, don't add any more amino acids to this chain. And it makes sense. If you're, doing anything that's in a chain, you have to stop the chain somewhere. Now, a nonsense variant, and I want you to kind of look right around here, is where one of these nucleotides, the C, is turned into a different one. So here a U gets put in. Now what that does is it changes that code. So instead of it having a C and encoding a star, now it has a UAA, which is a stop codon. And so it replaces that star and all the chain after it with a stop, okay? And that's called a nonsense. Now you might go, where did they get that? Well, it's to delineate it from the next type, which probably is what most of you guys got on your report. So a truncating frameshift variant is where you have, again, a nucleotide here, which gets deleted. And when it gets deleted, what happens is everything shifts over one, which means it gets out of that 3 nucleotide phase. So now it's out of phase. And when it does that, it changes everything. Now, the way things tend to work is it might add a few amino acids to the chain, which are incorrect. Like here, it was star, rhomboid, heart, moon. Now you've got pentagon, trapezoid, cross, and stuff. Okay, so that adds a few amino acids, and then usually there's a stop codon pretty close to the change. Okay, so if we're going to interpret the report, here it said, you know, at cDNA position 833, there was a deletion of a guanosine, and that caused a protein change at glycine 278 to an alanine, and then there were 4 amino acids placed inappropriate, so frame shift 4, and then X or asterisk means a stop codon. Okay? Now, the big question when it comes to these sort of changes are, you got one set of proteins that are coming from the gene that's on mom's chromosome, and then you got one set of proteins that's coming from the genes that are on dad's chromosome. Now, when I say dad's chromosome or mom's chromosome, realize most of these cases are de novo, so it's not on any of the parents' personal chromosomes, it's just on the one that the child inherited from them. And this is a normal dose, okay? Now, if the protein gets made and it's shortened, it could be that this protein doesn't work, okay? And it's 50% of the dose. Now, if you are somebody who likes to read on Google or something like that, we call this haploinsufficient. Haplo means half, insufficient means insufficient. So it's 50% sufficient. Now, the other thing that could happen is if this protein is made, it could actually inhibit the good protein's function. And then you've got less than half of a dose. And we typically call these sort of changes dominant negative ones, but you don't need to worry about that because we don't have those in GAND. Now, there's also a machinery in the cell that when you have one of these truncating variants, a nonsense or a frame shift, the cell actually senses it and gets rid of the messenger RNA. So the protein's never made, okay? And so again, you only make the good copy, but you're making the good copy in 50% of the amount you need, okay? And the way this works is something called nonsense-mediated decay. It should mean nonsense and frameshift-mediated decay. You guys don't need to know this, but it basically means if you have one of these, the messenger RNA of the bad copy gets destroyed, okay? This doesn't work at the very, very end of the gene, which we'll talk about maybe later in this talk, but tomorrow. But in any case, when we looked at cells from GAND patients and compared them to typical individuals, we saw that when we were looking at the messenger RNA, it was about 50% of the typical levels. And then when we looked at the protein, it was also a little bit around 50% as well. So we realized the protein wasn't made, so it has this model seems to work here. So the protein's not made, only the good copy's made, but it's at 50% dose. Now, some of you might say, well, if it's only the good protein, then why is there a problem? And then I like to use my analogy of building a building again. So say you're building a building and you have a pile of bricks and you go to start building the building and you don't realize at the beginning you don't have enough bricks. So if you only have 50% of the bricks, the building's not going to get finished. Now, alternatively, say if we go back to this one where the protein was made, that would be an analogy of you have a pile of bricks and half of them are bad and you build a building, the building's not stable, okay? But in reality, this is what's going on here. And so when we did this first study back in 2020, we had 50 patients, and they were 50 of the first 74 that were identified. And most of them were these nonsense and frame shift variants. And you can see they're right at the beginning. This is the gene here. This is the GAND gene with the exons and introns, and then this is the translated protein. And I'm using the protein as the map, but I really should be using the gene here. But in any case, you have these E77X. That means I think that's, I shouldn't have chose that one because I forget what E is, but say this D is aspartate. So aspartate 106 is changed to a glycine with a frame shift adding 5 amino acids and then a stop codon. And so these are all throughout the gene, but you'll notice that they're not really at this end here. And we're going to get into that later on. But basically all of these black lettered variants get destroyed. The messenger RNA gets destroyed and only the good copies made. So the next type of variant, which are these variants in green here, are what we call splice site variants. Now, we kind of skipped over this before, but when you go from the master blueprint to the working blueprint, what happens is it will make a copy of the gene with the introns and exons. And like, a cell is just a stupid cell. It has, it's like a bunch of robots working together. It can't say, oh, well, this is an intron and that's an exon. What it does is that there's sequences right after and right before the exons that say, hey, this is the good part, keep this, and get rid of the stuff in between. And the way it does that is that one of these sides binds some proteins that I'm not going to go into, recognizes the other side, and then loops out these sequences so that now all you have is the messenger RNA in its final form. Okay. Now, how do you have a splice site variant? Now, for those of you who got your genetic report back, and it will say something like C period 130 plus 2 G to C. What they're saying here is this is the code that recognizes this side of an exon, and this is the code that recognizes this side of an exon. And if you've got a change here, they say that's plus 2 to whatever this last nucleotide was there. So I'm going to give an example. So say there's a variant here. It's upstream of that, so it's a -2. Don't need to worry about the specifics of that, but if your genetic report says +2, +1, -1, +1, that sort of thing, it's going to be a splice site change. And what happens is this binds proteins, goes to recognize this, but because of this mutant, it skips it and jumps to the next code down the line, okay? And when it's making that messenger RNA, what happens is it excludes exon 5. And when it excludes exon 5, that's going to change that code, that nucleotide, the messenger RNA, so the code is off again. And it'll likely cause a frame shift. Now, the thing about splice site variants are these aren't like set in stone. So sometimes you can have a splice site variant where maybe 5% goes through correctly. The machinery, for some reason, recognizes it okay. And so if you've got 45% of that bad, 5% of that good, that totals that gene's 50%. And then when you add it with the good copy, you got a 55% level, which is more than 50%, okay? Now, sometimes when it comes to splice site, it's not that bad a variant, and so maybe 20% is made, and so your total amount then is 70%. And our hypothesis is that some of the children that are on the more mild side of the spectrum with GAND might have splice site variants that are allowing them to produce a little bit more of the good copies and the good protein and stuff. We've got a study that we're just finishing up to look at that. And of course, like I said before, that goes through nonsense mediated decay, the ones that don't get spliced correctly. Now, finally, we've got what's called GATA-D2B missense variants. And basically what happens is you have a change that's occurring here that's substituting the G for a C, changing the codon, and that changes one amino acid, okay? Now, if that amino acid is in an important place in the chain, that can cause disease. If it's not in an important place in the gene, it is a variant, but the genetic companies don't necessarily know if it's pathogenic, causing disorders, or if it's benign, okay? And that's where I come in. But you know, basically the idea behind missense variants are if you have a change at one part of the chain, and that could shut the whole thing down. And if that case, the protein's made, but maybe that copy doesn't interact with anybody. And so again, it's just like it's not even there. And so you have a 50% dose. The other alternative is you have that change, now something's being made, and it might decrease the activity of the other guy. And so you have a less than 50% dose. And we don't know much about this stuff right now, but you know, when we do look at those expression levels, the kids with missense variants, when it comes to their messenger RNA, it's the same as children who have typical GAN, but half of this messenger RNA is going to be bad. And the same thing for the protein. They're going to make the same amount of protein as the typical case, but half of it doesn't work. So, any case, those are the different types of variants out there. And once we look at this, you know, I'm getting reports from all over the world, and families are saying, hey, is this real or not? And I have to go through those reports and say, all right, now, what happens the most is these missense variants come to me, and the families are like, hey, my child has GAND, and then if you guys have emailed me, the first thing I ask you is send me the report and send me some photos, okay? And one of the reasons I do that is because a lot of the GAN children have a particular look to them, and if I get a picture where the child doesn't have that, my spidey sense kind of goes up and goes, maybe this isn't it. The second thing that happens is in the report, they're going to tell me whether the variant was inherited from mother or father. And that's a red flag to me, where if the parents are unaffected and the child is affected, but the parent has the same variant, that tells me that perhaps they have a change that doesn't do anything. And it's a missense change that's in a part of the gene that's not valuable. And we'll get into that a little bit more in detail and stuff. But I typically, if you're going to write me, I want to make sure we're not wasting each other's time by saying, somebody told you your child has GAN, your child doesn't look like GAN children, and your child inherited it from mom's not affected, probably somewhere else. And I've got examples that I'll show later on of where that happens. So in any case, Now let's get into GAND. So this is the first paper about patients with GAND where they actually focused on GAND. And it came out of Holland. And there was four cases. All the children had intellectual disability, infantile hypotonia, limited speech, and they had facial dysmorphisms, although these kids all look pretty to me. And they basically did some work where they looked at fruit flies just to show that the GATA-D2B protein was a little off with these cases, but the reality was it wasn't a really great place to look. But in any case, I looked at this paper, and my background is in gene expression, and the papers that were talking about GATA-D2B were all about gene expression, what makes a gene in the liver turn on versus in the brain. And I thought, this is something I'm kind of interested in. And in the meantime, the family in Los Angeles basically looked on Facebook and found about 5 families and said, hey, what's going on, and stuff. And they were trying to get this Dutch group to work on the disorder. And the Dutch group, they basically just identified disorders and move on. So they weren't interested in that. And so I went to them and said, hey, I'd be interested in working in this. And they were like, oh, we were going to ask you to work on it. And I was like, all right. So the first thing we did was we had to change the name, because at the time it was called autosomal dominant mental retardation type 18. We wanted to get something that had a little zing to it, so we decided GATA-D2B associated neurodevelopmental disorder, so GAND. GAND rhymes with hand or other things and stuff. And they formed the parental group with me working together. And what we decided to do was try and identify as many patients as we could at the time. And over the next 3 or 4 years, like I said, of the 74 that were out there, we got 50 to enroll. There was some in North America, South America, the UK, Europe, Australia, all over the place. And we wanted to find their genetics, how they presented, and their family histories. And as I said, the first thing we looked at were the genetics. And I've already shown you this picture with all the mutations. And most of them are these truncating mutations, or what we call loss of function mutations. And then, of course, there was about 8 that were splice site variants, and then there was, this is not from the paper, so at the time there was only 6 missense variants, okay? Now, the first thing you're gonna realize is that when you're looking at the splice site variants, they're always gonna occur where this white-gray border is. Now, not back there, but here, so if we look at this, follow the little line up, goes up to the border between exon 2 and exon 3, okay? And the same thing will happen here on the border of exon 5 and exon 4, exon 5 and exon 6. Okay, so then when we look, which makes sense, because that's where splice site variants are. But when we looked at the missense variants, we noticed that the missense variants clumped in one region or the other. And these 2 regions had already been identified by a German group, Dr. Renkowitz, as being conserved regions. So this is conserved region 1 and conserved region 2. Now, how do they get their name conserved region? Okay, when you look at GATA-D2B, there's another gene that humans have called GATA-D2A. Now, if you go back in time and you go to Drosophila, which are fruit flies, they only have one GATA-D2 protein, and it's called Simjang. But from there, it divides when you get into fish and lizards and move up the vertebrate scale. So conserved region 1 and conserved region 2 are called that because when you look at GATA-D2A, the same regions are very similar, okay? Where the rest of the gene is pretty variable between the 2. Now, if I go through this and look at a human GATA-D2B, and then a dog, and then a chicken, and then a frog, and then a fish, they slowly get a little bit less similar, but they still stay pretty similar. And that tells me what are the important parts and what aren't the important parts. And I'll show you a little bit of that tomorrow. But these missense variants being in these 2 regions told me, hey, these are important regions where maybe these other areas are not that important, okay? And we're gonna get into that in a little bit. And that's all of them together. So at the time, we had 33% were nonsense and frame shift. Well, 33 of them were, which was 66% because we had 50. We thought 7 were missense and 7 were splice site and 3 were deletions, but we subsequently realized that this one here, we thought was missense, is actually a splice site, so we've changed that. So it's actually 6 missense and 8 splice sites. Now, one of the things back then was that when we looked at the kids with those 6 kids with missense variants, 4 of them had seizures, which is 66%. Or when we looked at all the rest of the kids, only about 25% had seizures. And so one of the things that we'll talk about tomorrow is over the last 3 or 4 years, I've been collecting missense variants to see if when I get those numbers up to around 30, does that hold true and stuff. But in any case, so we had 50 kids. Most of them were these nonsense frame shifts, but some of them were the opposite ones. But that told us a lot already. We learned a large amount. When we looked at the 50 kids, you know, these were the 3 things that the Dutch group saw: infantile hypotonia, motor delay, intellectual disability, and distinct facial appearance. We figured out that a lot of these kids, about 80, 90% of them, had large heads. And then we also noticed that many of them had this oral mechanical dysfunction, which is in pregnancy, they had a large amniotic sac because they weren't swallowing the fluid. When they were born, they also had this oral mechanical problem with feeding, so neonatal feeding issues. And then when they got older, they had apraxia of speech, meaning not only did they have the intellectual disability cause issues with language, but the actual use of the mechanical part of producing speech was also having problems. About 24% had epilepsy, but like I said, when it came to the missense, about 60% did, and about 60% of them had abnormal brain MRIs, although the MRIs of the brain were not tremendously abnormal. And then, like I said, about 8 to 10% had these aortic valves that were bicuspid. And that means that instead of looking like normally it's a tricuspid valve that looks like a peace sign. So when the heart pushes blood out, that valve opens up and lets the blood go out and then holds it from regurgitating back into the heart. And a bicuspid valve, you just have 2 valves that open up. And what can happen is where those valves kind of finish, you can get calcifications and things that can slowly make that valve close off. And that's one of the things that if your child is diagnosed with a bicuspid aortic valve, the cardiologist will probably want to follow that over time to make sure that doesn't get smaller to the point where the heart can't push the blood out. And so it has to get bigger and bigger and bigger, which can cause problems with the heart's function and electrical conduction. In any case, the children also had some facial characteristics. This is from our first paper in 2020. I'm not sure if anybody's child is on here or not, but again, we had high wide forehead, frontal bossing, ocular hypertelorism. This is a program that puts together kind of an image of what's going on. And they had this same nose. They had a short philtrum, which is the space in between your nose and your upper lip. And they all have a pointed chin. And when you look at the kids, you can see they all look like brothers and sisters because they all have kind of these same sort of features and stuff. Now, the things that we found in this group, you know, about 42% had that triad of kind of oral mechanical dysfunction with polyhydramnios and 42%, neonatal feeding issues in 82%, and then apraxia of speech should be in here at 100%. Intellectual disability was in 100% of the children, with about 20% being nonverbal after 4 years. And when I say nonverbal, meaning no words. And I already mentioned the seizures and such. And then, you know, there was about 5% that were non-ambulatory after 3 years. Almost all the children had good eye contact and social reciprocity. I don't like to use the word autism in my research anymore because autism has changed in its definition over time, so that one child being autistic, the whole point of having nomenclature is when I say a child is autistic, you can imagine what that child looks like. But now you really can't do that. So I use good eye contact and social reciprocity, meaning when something good happens, they go to their parent or they want to share that good with other people. One of the big things a lot of families ask me about is toilet training. And, over 4 years of age, really about 63% are still not toilet trained. And it's one of the big hiccups that can occur here. I tell parents, of 11 and 12-year-olds, keep trying, but if it's not working, do not beat yourself up. It might just not happen. But it's one of the things that most families want to know about and everything. Again, about 8 to 10% with the bicuspid aortic valve. 88% had strabismus, which is, we call it a lazy eye in the United States. I think you call it a squint here. And most of these children should be seeing an ophthalmologist as well. And then we already talked about the facial appearances. So right now, where are we at? Well, I say greater than 500. I think we just got over 500 recently and stuff. A lot of what we're learning is because we've done the clinical work. Now, I was just in a meeting in Barcelona called the Chromatin Structure and Function Gordon Research Conference. And there's a guy from Exeter and a guy from Sydney, Australia, along with my collaborator in Miami. And then there's another guy, I think, on the East Coast of the United States in North Carolina. We're the 5 people in the world that really work on this nucleosome remodeling and deacetylase complex. And so I finally got to meet, there's another person at Exeter. So I finally got to meet 3 out of the 5 or 6 and stuff. So that's why I went to that meeting, just because we're very isolated. And I'm going to explain to you why people don't work with NERD and how the clinical research is actually starting to wake some people up when it comes to this. So, some things about GAND, so let's talk about GATA-D2B. Just need a little break. So, this is so that if you're looking at papers or you're on Google and stuff, if you see GATA-D2B written in all italics and all caps, they're talking about the human gene. If it's in capitalized at the beginning and in lowercase, but all italics, it's the mouse gene. So italics equals gene, all caps equals human. If you have all caps, but it's not italicized, we're talking about the protein. Because the reason you might go, oh my God, why didn't they just give it a different name? Well, they did that with a lot of other diseases. And what happens is you have GATA-D2B encodes P66 beta, and then people don't know that P66 beta is associated with GATA-D2B. So we just do it this way. The problem is when I'm speaking, sometimes I have to say the protein or the gene, because I can't speak in italics or non-italics. But in any case, the mouse protein has all capital in the beginning and all lowercase letters and stuff. So this is the NERD complex here. And what you have is you have 2 big parts. One is called the histone deacetylase core, and one is called the nucleosome remodeling core. And that's where it gets its name, nucleosome remodeling and deacetylase complex. Now, this histone deacetylase core is actually promiscuous. It can bind with other subcomplexes. So this is really the nucleosome remodeling. That is the essence of NERD, okay? So these 3 proteins can get exchanged with the HDAC core and form a different total complex. So this is where it becomes important. Now, and this is where the only disorders are located when it comes to NERD. Now, so you have basically a methyl binding protein, a methyl binding domain protein, GATA-D2B, and these CHD4 or CHD proteins. Now, the CHD proteins are the important proteins. They're the one that do the chromatin remodeling. but GATA-D2B and GATA-D2A hold it in place. So if you don't have 2A or 2B, you don't have the CHD proteins, and that's important. But in any case, we're going to get back to this. So what did I just call NERD? The nucleosome remodeling. And when we were showing this, I was telling you, you have DNA wrapped around proteins that makes a nucleosome. So that's where we're going now. So this is what it looks like. The colleague in Australia provided this. And this here, you can see is a piece of DNA wrapped around a protein. So that's the nucleosome. And then this is NERD. Now, GATA-D2B is this gold protein here. And what you can kind of see is that this is the HDAC core, and GATA-D2B is holding the CHD protein in place with that, okay? Now, NERD, because it's working with nucleosomes, remember I said the more nucleosomes, the more closed the area is, the less nucleosomes, the more open it is. If it's open, the genes can be expressed. If it's closed, the genes get repressed, okay? And that's really important when you differentiate stem cells from a stem cell into a neural progenitor and then a neuron, or a heart progenitor and a heart cell. And neurogenesis is basically the process of making neurons in the brain. And then cerebral cortex development, or corticogenesis, is making the complex structure of the outer part of the brain, or the cortex. So basically what GATA-D2B is doing is tethering the HDAC core into the CHD protein, okay? And that's important because in those areas where it's interacting with the MBD protein, that's the CR1. And where it's interacting with the CHD protein, that's this GATA-zinc finger in CR2. And that's why they're conserved. You need to have the amino acid sequences in both of these areas to be very particular, kind of like Legos that only fit with other Legos. And if you change those, it's not going to bind to the HDAC core with CR1, or it's not going to bind to the CHD protein with this end. So just to kind of show it with this structure, these CR1 changes are what interacts with the MBD, and the CR2 interacts with the CHDs, okay? So that's why just by doing this clinical study, we showed that these missense variants are in an important region. And, you know, I basically called up Joel Mackay, who's down in Sydney, who made this structure. And I said, hey, would you want to do some collaborations with some kids that have these missense variants? And he was like, well, that would be exactly what I'd want to do. I've been waiting for that. And so within a month, he'd done them, and we published this paper and stuff. So if you thought that was complicated, it gets even more complicated, because at each one of these positions, you can have more than one protein. And we call like GATA-D2A and GATA-D2B paralogs, okay? So that means they're very similar and they do very similar things, but they're not exactly the same, okay? So at any given time, you can have GATA-D2A in this place or GATA-D2B, but you can't have both. The CHDs have CHD3, 4, and 5. So at any given time, you can have one of those, but you can't have the others. So they're mutually exclusive. And this is why people didn't do research on GAND or on NERD, is because you can form a whole bunch of different NERD complex subtypes. So how do you study that? It's not easy, because if you take out CHD4, well, then you still have CHD5 and 3 around and stuff like that. And I'm going to get into why GAND and GARD, the GATA-D2A disease, are very important when it comes to understanding NERD function. So basically, we published this paper about GAND. And the big things we said were macrocephaly, intellectual disability, motor delays, apraxia of speech, bicuspid aortic valves, epilepsy, and unique facial appearances. So then when we looked at the kids that had CHD4 related syndrome, where this is the CHD4, here's GATA-D2B, they had macrocephaly, intellectual disability, motor delays. They also had heart defects, but they were much bigger heart defects. The kids with GAND tend to have valvular heart defects, and there's a reason for that. We'll get into it. And the CHD4 kids had male genital anomalies as well as deafness, which are two things that really we haven't seen much in the GAND kids and stuff. Now, when we looked at CHD5 related syndrome, again, this is CHD5 sitting here now instead of CHD3 or 4. The kids didn't have macrocephaly, they had craniosynostosis, meaning the sutures of their skull were fusing early, okay? And they had intellectual disability, motor delays. They had some language issues, but their big thing was epilepsy, okay? And then when we looked at kids with CHD3, these are all being published around the same time. The kids who had variants in CHD3 had macrocephaly, intellectual disability, motor delays, apraxia of speech. They also had male genital anomalies, as well as a unique facial appearance that's different from GAND and all the other CHDs. And so, if we were going to put this together, imagine this is the HDAC core, and this is GATA-D2B, and this is all of the problems we see in GAND. If we then add the CHD4, we basically have the heart defects as well as all of these issues, but they also have severe heart defects, deafness, and male genital anomalies. If we add CHD5, they have all of these with epilepsy. And then if we add CHD3, they have all of these with apraxia of speech as well as the male genital anomalies and inguinal hernias and stuff. And so what you can see here is that for most of the neurologic problems going on with nerd-related illnesses, there's separate ones for each one of these CHD proteins, but GAND has all of them together. So the CHD proteins have overlapping presentations where GAND is a pan disorder of all of those. And so I call GAND a pan-nerdopathy, okay? And a nerdopathy is just any disease associated with nerd, all right? Now, why does that happen? Well, I kind of showed you that if you have if you don't have enough GATA-D2B, you're not going to bind the CHD proteins. And so if you have 50% of GATA-D2B, you're going to have 50% of CHD4 binding when it needs to bind, 50% of CHD5, and 50% of CHD3. And so, when I was at this meeting in Barcelona and I said, well, if you have this, you can study NERD because all the CHDs are now taken out together instead of just one of them. Because what people were doing was they were looking at CHD4 alone or CHD3 alone or CHD5. And they were getting different things. And I was like, well, this way we just get rid of all of them and stuff. Now, this only works if GATA-D2B is the predominant paralog during neurodevelopment instead of GATA-D2A, which we've proven that that's the case. So basically, that's why you have these presentations that, although they overlap with the CHD proteins, they're all present in the GAN proteins. So what we then decided to do was, like I said, we had about 60% of the missense cases that were having seizures compared to the other ones. And so what we wanted to do was do another study where we were collecting only missense splice site or intragenic deletions. And many of you probably got emails that said, hey, I'm really interested in your child, but I'm only interested in these particular mutations. And it wasn't because your child was less important, it was because these guys were the pieces that we didn't have from the first study. And so with the second study, we got a whole bunch more, took us 4 years instead of 2 or 3, and we got a bunch of missense variants, and again, they're located in this GATA zinc finger here. We got more missense variants here that are in the CR1 region, but then we started getting a couple variants that are a little bit away from the zinc finger there, and then we got a couple variants that are upstream here. Now, the interesting thing is when we look at GATA-D2A, this region here is also conserved. It's smaller than CR1 and CR2, but it's still conserved. And the weird thing is this aspartate 102 and this aspartate 124, they're like the core of what's conserved. So that tells me this is doing something. So my model before was this is a piece of Velcro, this is a piece of Velcro holding HDAC and CHDs in place. But then this has to be doing something. Now it might be stabilizing those other interactions, or it might be binding something else. Now, the big thing that I wanted to show was that now we had 33%, not 33, we had 33 missense total. We have 27 splice site total and 8 deletions. And when we looked at the missense kids, they had about 25 to 30% having epilepsy. So that increased epilepsy risk was just a function of having so few kids then. Now, the other thing you might notice is there's a bunch of missense variants up here in red and over here in red. And those are the kids that parents sent me reports. And when I looked at their pictures, I was like, they don't really have a GAND appearance. And then when I went through the reports, I found one of the parents had the variant themselves and passed it on. And so this tells me that these are benign variants. But it also tells me that this area here is probably not as important as we think. Now, there might be areas in here that are important, but because this is off over like 60 amino acids, probably means that section's not important. But in GATA-D2A, there's actually a different sequence there that is really important. So it kind of tells us maybe GATA-D2B evolved by getting rid of that important region that was in GATA-D2A. In any case, our next study, we're looking for kids with polygenic deletions, which involve GATA-D2B and other genes. We're also looking into parental mosaicism, and this is where one of the parents has had a mutation early in their embryogenesis, and so they can actually, it occurs in maybe 10 to 20% of their cells, of which some of those cells are in the ovary or testis and stuff, where they're normal, but they're passing this on. And then that might make you guys go, wait a second, if that's the case, then why couldn't that be here? And that's because those parents had 100%, where these other parents have 20% of their sequences or 5% of their sequences having the variant. But we'll go into that a little bit tomorrow. You know, what we're also starting to branch out in is looking at other NERD-related disorders. So there's this disorder called SAPB2 associated disorder, which we're going to talk about tomorrow because it plays into all this. But kids with SAPB2 associated disorder have intellectual disability, motor delays, apraxia of speech, seizures, and then they have some dental anomalies. Then there's kids who have disorders with FOXP1 or FOXP2, where we know FOXP1 and FOXP2 interact with GATA-D2B, and these children have apraxia of speech. So they can be completely typical, except they can't speak, or they can have intellectual disability or motor delays and things like that. And one of the research things that we're kind of going along here is we're noticing like, hey, wait a second, when we look at the NERD complex and it binding SAPB2 on the DNA, you got CHD3, that has apraxia of speech, FOXP1, 2, that has apraxia of speech, GATA-D2B, that has apraxia of speech, and SAPB2, that has apraxia of speech. So some of the research we're looking into is which cells in the brain are expressing all these proteins together. And we'll talk a little bit more about that tomorrow. Now, that's my last slide. So you can all take a breather and go, okay, this was really, my head was starting to spin, everything, I don't know what, I got lost. I'll just to give you guys some perspective, this chromatin structure and function meeting I was at in Barcelona, I would go to the talks, and my goal was, if a talk was 20 minutes, I could stick in it for 10 minutes. And a lot of the times after 5 minutes, I was like, okay, I'm gone. I'm going to work on some emails and stuff like that. So it's all a matter of where you're at and stuff like that. And hopefully some of the people who were here last year are like, you know what, now I get it a little bit more. And some of the new people are like, okay, that was way over my head, but it's one of the things we can talk about a little bit more later on. In any case, this is a picture from our last meeting here, and I want to acknowledge Rebecca and everybody involved. The biggest thing that happened between then and now is we had a lot of people here get nice and organized and send me those photos and reports. And then we also had a British woman who lives in Colorado who works with Helping Hands for GAND. Her name's Abby Taylor. She got involved and she started, because they're getting every single case in the world. People are contacting them and they just don't know what to do with a lot of them. And she sat down and she said, okay, this is a missense. This is a loss of function. This is a splice site, and started sending me those. So, she would send them an email to contact me and stuff like that, because we're very good about protecting your privacy and stuff. But we were able to close off the gap with that second study, and we're about to put that all together and publish it. This is my team at the lab. This is Halloween, and Gabby is someone who's done most of the work here. Phil Kenny, Maggie, and Haral did a ton of work. Who's not on the list is Molly, Eric, and Derek. I'll show you them tomorrow. And the nice thing that happens when you have family research groups like GAND UK and Helping Hands for GAND is they can fund our work so that we can get the preliminary stuff that when we write grants, they get impressed and they fund us. So about a year and a half ago, maybe a little more, California Institute of Regenerative Medicine awarded Dr. Young, who's our mouse guy that you'll hear more about tomorrow, and I a grant for about $1.4 million, which will run out a year from now, and so I need to get more money. But I'm writing a grant right now to our National Institutes of Health. I've been funded by the Fashion Industries Guild at Cedars-Sinai for a long time, and as well as the Board of Governors. And of course, I always want to thank Mindy, who's now the Helping Hands for GAND president, and her family, and Olivia here, who's that first patient, who's now almost a teenager, I think.